This video is going to deal with the beginning of what is called microbial metabolism. Microbes, just like human beings, they have to have food for energy, and this briefly covers and talks about enzymes and how do microbes break down a substrate. A lot of times it could be sugar, other things, and convert that into energy. On this section, there is a PowerPoint, there's an outline for study. This class outline was one that was used in a class that actually has blanks in it. So all the information is right there for the outline. And then here's the exam for this, what will be the second exam. It's over genetics and micrometabolism. So right here at the beginning, it says, how do pathogens acquire energy and nutrients at the expense of a patient's health? Well, if you're talking about necrotizing fasciitis, that's Streptococcus pyogenes. In other words, that strep bacteria actually uses a person's skin to break it down and convert it into energy. There are yeast that produce alcohol as a byproduct of fermentation. That's where alcohol comes from. And so I'm trying to make these videos short. All the information you need is in these outlines and the PowerPoints. And there is a lesson that deals with this as well. Hopefully this will let me go backwards. Yeah, right here under this assignment for this unit. This is lesson 3, and I'm going to add that. But here we go. And it does kind of mirror, and these are questions based on that, but it does actually talk about enzymes and this part we're about to get to here. Okay, so 8 statements about metabolism. Every cell acquires nutrients, which are chemicals that are necessary for building blocks and energy. Energy is stored in the form of chemical bonds of adenosine triphosphate. Yes, since we're talking about microbes, if you're using oxygen, if they are using oxygen, then oxygen is used to convert food energy into ATP. If they are an anaerobe without oxygen, they're doing a process called fermentation, which is not using oxygen directly to convert a substrate into ATP. Using enzymes and energy from ATP, cells construct larger building blocks. That's anabolic. If you're talking about tearing something down, that's catabolic. Stuff drives me crazy. Okay. So, yeah, there are times when a cell has to build up something, and then there's times that are catabolic when they have to break down a substrate and get energy from that. So I'm going to go down here to this little diagram. So here is the role that enzymes play in metabolism. This diagram is really easy to follow. It's showing here this big arc and it comes down. Now, every chemical reaction, there are reactants and there are products. Products are the result of the chemical reactions involving the reactants. All chemical reactions, and that's what this is talking about, they require an activation energy. That is the amount of energy needed for that reaction to occur. Enzymes act as catalysts. That's why most chemical reactions will not occur unless there's an enzyme present. And the reason is that, see this arc, that amount of energy is usually too much for the reactants to overcome. So this dotted line is showing when you have an enzyme involved with a chemical reaction, there's much less energy. So basically, enzymes lower what is called the activation energy for a chemical reaction to take place. In bold right here, a catalyst, as stated earlier, is a chemical that starts chemical reactions. Enzymes lower the activation energy required for a chemical reaction to take place. So on to the next part. What's an enzyme? Well, an enzyme is it's held together by bonds, and you really can't see them. Now this, on our lesson, I just saw that. Look at that craziness, and this is 2 of them joined together. That's an enzyme. See all the folds and everything? And here's the thing. Enzymes have a certain shape, and if that shape is changed, the term is denatured, then the enzyme is rendered, is not functional. But yeah, all these twists together, that's an enzyme. And they're held together by chemical bonds. So here's a much simpler diagram. See the enzyme here? Now, the area called the active site, it's actually trying to point to this and also that. But like I said, enzymes have a certain shape. Now, a substrate is the compound in which enzymes act upon. All enzymes have a certain shape. Part of the enzyme is called the active site. It's actually a binding site, and that's the part where it binds to its substrate. Now, if you're asking me, what is a substrate? Okay, I'm going to give you a human being example. Sucrose, table sugar, found in candy. It's a disaccharide. Sucrose is too large to be absorbed in the small intestine. So, human beings, animals, we have an enzyme called sucrase that's located in the small intestine. When sucrase, which would be the enzyme, comes in contact with sucrose, sucrase allows a chemical reaction to take place that breaks sucrose into two monosaccharides, two smaller units, that can then be absorbed. So that's the role of enzymes. And you do have enzymes that are anabolic, they build up, and you have enzymes that are catabolic, that actually break into smaller pieces. Yes, and the term denature means if the enzyme structure is altered, then the enzyme is no longer functional. So we'll cover that in another video. So, enzyme activity. Here's an example, and remember, these are all reversible. So here's the enzyme. Here are two substrate molecules. You see the enzyme is starting a reaction in which these two smaller units are then joined together to make a larger one. This is an anabolic reaction. There are enzymes like this one here. And the same enzyme. Okay, and enzymes are not really affected during a reaction. They can be reused, but if this one were to bind here, there are reactions that allow it to go the other direction. So it's taking this larger molecule, this is catabolic, and splitting it into two smaller ones. That was my sucrase example, or sucrose. There are bacteria that can ferment lactose. Lactose is a milk sugar. Lactose is a disaccharide. Bacteria that use that for energy, they have enzymes that take this and split it into two monosaccharides, simple sugars, and then that cell can then use those for energy. So the active site, I showed you that. The active site is the area where it binds to its substrate. And a substrate is the compound of which an enzyme acts upon. Okay, I'm going to start the next video here.